Good afternoon, everyone. Now, eyes are essential and special sense organs which give us the wonderful sense of sight. Who could imagine that this small space could contain the images of the universe? I will be dealing with the following topics. The following are the topics that I will be covering today: physiology of vision, visual pathway and field effects, binocular vision, and a few electrophysiological tests, namely the electrooculogram, electroretinogram, and the visually evoked potentials. Let's start with the physiology of vision. Have you ever wondered or asked yourself this question? How do we see what we see? Well, light is a form of energy whose interaction with the retina gives the sensation of sight. Light is composed of different wavelengths or colors, and white light is a combination of all of them. As you can see, this is the electromagnetic spectrum, and the visible spectrum is between 723 nanometers at the red end and 397 nanometers at the violet end. So, we can see light only in this part of the spectrum. Retina. The retina is the photosensitive part of the eye. Rod and cone cells in the photoreceptor layer transform light stimulus into a nerve impulse. Impulses are conducted by the nerve fiber layer of the retina through the optic nerve and ultimately to the occipital visual cortex in the brain. As you all know, the retina consists of 10 layers. Let's revise these layers once more. Let's start from the outer to the inner. We have the pigment epithelium, the layer of rods and cones, the external limiting membrane, the outer nuclear layer, the outer plexiform layer, the inner nuclear layer, inner plexiform layer, the ganglion cell layer, nerve fiber layer, and the inner limiting membrane. Now we can broadly divide the 10 layers into two regions. That is, the outer region is formed by the retinal pigment epithelium, and the inner 9 layers, this is going to constitute the neurosensory retina, which is responsible for the visual impulse. The retinal pigment epithelium, which is the outermost, has a few metabolic functions of its own. There's one more point I want you all to remember. The axons of the ganglion cell layer form the optic nerve. Now, what are photoreceptors? They are namely cones and rods. Cones. We have 6 million cones in each eye. They are sensitive to bright light and color, and they are responsible for vision during the daytime. The pigment found in cones is iridoxine. In the fovea, which is situated in the posterior pole of the eye, only cones are present. Rods. We have 12 million rods in each eye. See, there's a huge difference in number. So rods are more in number than the cones. This is stimulated by dim light, and they help us seeing during the night time or for scotopic vision. The pigment that is found in rods is rhodopsin. Rods are more concentrated towards the periphery of the retina. Physiology of vision. Now let's see 3 steps that involved in the physiology of vision. So step 1 is initiation of vision by a process called phototransduction that takes place in the rods and cones. The second step would be processing and transmission of visual sensation, which is a function of image processing cells of retina and the visual pathway. The third step would be visual perception, function of visual cortex and related areas of the cerebral cortex in the brain. We have 2 important changes happening, when we see. When parallel rays of light fall on the retina, there's going to be a photochemical change occurring in the photoreceptors, namely the rods and cones. The chromophore present on these photoreceptors absorb light and form a series of intermediate substances with the help of vitamin A. This starts the visual process by changing the electrical potential transmitted to the brain via the neurons. The electrical changes. Now light is going to stimulate the photoreceptors in the retina, which sends electrical impulses through neuronal network in the retina and the visual pathway to reach the visual cortex. So, electrical changes is mainly the conduction of electrical impulses to the brain. What is nyctalopia or night blindness? It is one of the first signs of vitamin E deficiency. Remember in the previous slide I told you we need vitamin A for photochemical changes? So a deficiency in vitamin A reduces the production of rhodopsin, which is a pigment found in rods needed to see in low light. Treatment would be supplementation of vitamin A. Causes of night blindness include retinitis pigmentosa, uncorrected refractive error, myopia, cataract, congenital stationary night blindness, and advanced glaucoma. Here's a question for you. Cone cells are photoreceptors that are seen in A. Ora serrata region, B. Colored light, C. Fovea, and D. Vascular tunic. Can you answer this? Fovea. Now, cone cells are densely packed in the fovea, and they quickly reduce in number towards the periphery of the retina. Here's another question. The rods help in scotopic vision. State whether true or false. Yes, correct answer. True. They're responsible for night vision, but rods cannot differentiate colors. Colors are a function of cones. Visual perceptions. These are sensations which result from stimulation of the retina by light, and there are four kinds: light sense, form sense, sense of contrast, and color sense. Light sense. If the light falling on the retina is gradually reduced in intensity, there comes a point when it is no longer perceived. This is called as light minimum. The eye functions in a wide range of lighting conditions, and adaptation to such changes is necessarily very rapid in daily life. We are all aware that if we go from bright sunshine into a dimly lit room, we cannot perceive the objects in the room until some time has elapsed. The eyes have to be adapted to the amount of illumination. The rods are more sensitive to low illumination than the cones. So in the night, we see with our rods, and in bright illumination, we see with our cones. And in twilight, for mesopic vision, both rods and cones come into play. Form sense. It is a faculty which enables us to perceive the shape of objects. Here, cones play a predominant part, and form sense is most acute at the fovea, where the cones are closely set and most highly differentiated. Visual acuity is the capacity to see fine details of objects in the visual field. The acuity of vision, therefore, applies to central vision, that is, images formed at the fovea. Now, visual acuity is a record of form sense. Sense of contrast. The ability to perceive slight changes in luminance between the regions which are not separated by different borders. In this picture, you can see there is a difference in contrast between the two images. Now, a test with Pelly-Robson chart can identify defects in sense of contrast. Color sense. Color vision is the ability to distinguish between different colors as excited by light of different wavelengths. Normal color vision is trichromatic, red, green, and blue. It is a function of the cones and occurs only in photopic vision, that is, in bright light. It can be assessed by a simple test using the Ishihara pseudo-isochromatic plates. Why is color sense important? In optic nerve involvement, there is a preferential inability to distinguish between red and green colors. In macular abnormalities, there will be a preferential loss of ability to differentiate between blue and yellow colors. It is important to test color vision in certain occupations like drivers, pilots, sailors, and in armed forces. Visual pathway and field effects. Visual pathway. It is a pathway of conduction of visual impulses from the retina to the visual cortex. It consists of the optic nerves. So we have two optic nerves. These optic nerves meet at the optic chiasma. Then we have a pair of optic tracts, ganglion cell, lateral geniculate bodies, optic radiations, and the occipital cortex. So there are 6 components of the visual pathway: the optic nerves, optic chiasma, optic tracts, lateral geniculate bodies, optic radiation, and occipital cortex. The visual pathway. Light from the temporal field is projected on the nasal retina. Light from the temporal visual field is projected on the nasal retina. And light from the nasal visual field is projected on the temporal retina. Light from the nasal visual field is projected on the temporal retina. So they're going to be opposite. Axons of the ganglion cells from the optic nerve leave the eye and pass at the optic chiasma. Remember I told you axons of the ganglion cells, they're going to form the optic nerve. At the optic chiasma, fibers from the nasal halves of the retina cross towards the opposite side, whereas the temporal fibers remain uncrossed. The fibers from the optic tract end in the lateral geniculate body. From the lateral geniculate body, the fibers pass by the way of the optic radiations to the primary visual cortex in the occipital lobe. The messages which are transmitted in the form of visual impulses from the two eyes are integrated in the visual cortex to create a single image seen upright. Any lesions of the visual pathway can cause defects in the visual fields and diminution of visual acuity depending on the site of lesion. Here's a question for you. The electrical signals from both eyes merge at the A. optic nerve, B. optic chiasm, C. lateral geniculate bodies, D. the striate cortex. Can you please answer this question? Your answer is correct. It is the optic chiasm. It is the part of the brain where optic nerves partially cross. Now let's see what binocular vision means. Binocular vision can be defined as the fusion of the sensation produced by light stimulus into each eye as a unified perception. There are a few requisites for binocular vision. That is, the optical apparatus should be approximately equal in both the eyes. Visual fields of the two eyes should overlap. Visual pathway should be uninterrupted. And there should be proper extraocular muscle coordination. When a person looks at an object, the light rays coming from the objects stimulate the retinas of both the eyes. The input from both retinas get fused at the visual cortex so that the brain perceives them as a single image called binocular vision. There are three grades of binocular vision, that is, simultaneous perception, fusion, and stereopsis. Simultaneous perception, fusion, and stereopsis. Now, let's just go on to basics of the electrophysiological tests. Let's remember few basic points that will help you recall these tests whenever you need them. Now, electrooculogram is going to record the function of the retinal pigment epithelium. The electroretinogram is going to tell us about the function of the photoreceptor layer. The visual evoked potential is going to tell us about the functioning of the ganglion cell layer and above. Electrooculography. It records the effect of light and dark adaptation on the resting potential generated at the retinal pigment epithelium. The retinal resting potential is 6 millivolts across the retinal pigment epithelium. The receptor side is electrically positive and the choroidal side is negative. It is used in the diagnosis of retinal dystrophies, retinitis pigmentosa. Electroretinography. It records the electrical activities of the retina by stimulating it with a flash of light. It gives information about the functional integrity of the retina. There are three waves in the ERG. A wave is from the retinal photoreceptors, B wave is from the bipolar cells, and C wave is from the pigment epithelium. ERG is helpful in the diagnosis of retinal detachment, retinal dystrophy, and vitamin A deficiency. Visual evoked potentials. VEPs are electrical potentials recorded using scalp electrodes over the occipital cortex of the brain on presenting visual stimuli to the subject. VEP waveforms are N75, P100, and N145. So, we measure the latency, duration, and amplitude of P100 wave. Abnormalities in the latency and amplitude are helpful in diagnosis of diseases like optic neuritis, optic atrophy, refractive errors, and color blindness. So I'll be ending my talk. Thank you.